Behold the nucleus and sidekick dots, codenamed ribosomes. I am going to tell you the story about a very famous man named Nucleus. The nucleus controlled when the cell produced what proteins, when, where, and how we controlled growth, and regulated metabolism. But basically, the nucleus wanted cell domination. Once upon a time, there were bazillions of nucleuses, one for each eukaryotic cell. It had a double membrane consisting of 2 lipid bilayers, and the whole organelle measured just 5 U with the tail thingy M's in diameter. But the nucleus wasn't all smooth. It was covered in pores, and each 100 nanometer diameter pore was fused, opening only to regulate the entrance and exit of certain particles and macromolecules. The inside layer of the nucleus's membrane was lined with a nuclear lamina. The lamina is a net of protein filaments which maintain the eggish nucleus shape. Well, the nucleus had chromatin, a purse which he had kept his DNA and protein in. If the nucleus ever decided to get divorced and have kids, the chromatin purse would enlarge from being stretched between the divorcees, and you'd be able to see the money. Chromosomes. One day, the nucleus controlled protein synthesis, like he did every day, by sending a chemical messenger named ribonucleic acid bottom, or mRNA, to the DNA. The DNA synthesized the acid bottom, and acid bottom conveyed a secret message to the cytoplasm through the ninja nuclear pores. Then, acid bottom attached himself, several of himself, to ribosomes, where the secret message was decoded and made into the primary structure of a specific protein. Thus did the nucleus achieve cell domination. But, dear me, I've almost forgotten about the lonely little ribosomes. Now, earlier, we called ribosomes the sidekick of nucleus, and that's because the nucleus needs Ribby in order to make proteins, and generally be the henchman in cell domination. One kind of ribosome is called free because it, or they, floats around in the cell cytoplasm, where the ribosome's protein soldiers mostly function in the cytosol, such as enzymes which catalyze metabolic processes. Enslaved, or bound, ribosomes are doomed for either membrane inclusion, packaging, or export. However, whether he be free or bound, all ribosomes are the same at heart, structurally identical and interchangeable, and the cell can adjust the relative number inside of the cell.